This video is all about the nucleus. So let's start off by looking at the animal cell that you'll recognise from GCSE. So we often, at this level, draw an animal cell looking a little bit like a fried egg. So you can see the outer cell membrane, you can see the cytoplasm, and you can see some mitochondria within the cytoplasm. You might also know about ribosomes from GCSE. Now, they're not on this diagram, but you know that they're present too, a little smaller than the mitochondria. But what we're interested in is the nucleus. So as you can see here, up to GCSE level, you often just see the nucleus as a blob in the center of this cell that looks like the yolk of a fried egg, if you like. Well, at A level, the nucleus looks a little different to this, and it has lots of different components that you actually need to know the role of within the nucleus and its function. So again, at GCSE, this is what you're taught, okay? You're taught that the nucleus contains the genetic material, which includes the DNA, which controls the cell's activities. Okay? So this is not false, this is true. The nucleus does control the cell's activities, but there's a little bit more to it than that. And at A level, you'll learn the extra detail about the nucleus and all of its different components and their role too. So this is what a nucleus would look like at A level. Okay, so can you see there's a little bit more detail that's being revealed here? And I should point out as well that this is also highlighting one of the skills that you need to learn in A-level biology, which is about how to produce biological drawings. So obviously this is an artist's impression of what the nucleus looks like, which is taken from a photograph that somebody will have taken of this cell under an electron microscope, because this sort of detail you can't see with a light microscope. So with a light microscope, you would just see a dark shadow where the nucleus was. But this sort of detail is revealed when we use an electron microscope. And electron microscopes allow you to see this detail because they have both a higher magnification and a higher resolution. It's actually the resolution that allows you to see the detail and not the magnification. So a higher resolution allows you to see all of this detail that you couldn't see with a light microscope, because light microscopes have a lower resolution. Okay, so let's see this extra detail that we can view with this electron microscope. So first of all, around the nucleus, you have a membrane. And you can either call this the nuclear envelope, as we have here, or the nuclear membrane. It doesn't matter which, okay, but we'll call it the nuclear envelope. Okay? Now, within the envelope are little holes, okay, and these are called pores. So the envelope has gaps in it. And then you can see these little squiggly lines inside. Now, that's something that we call chromatin. And that's actually the DNA. Now, inside of the nucleus is also a jelly-like substance, very similar to the cytoplasm that you know within a cell. So the nucleoplasm is the jelly-like substance that all the DNA floats around in. And then can you see there's this big dark blob in the center? So this is a structure called the nucleolus. So let's take a look at the function of all of these different parts of the nucleus. Okay, so the nuclear envelope, like I said before, is a membrane that surrounds the outside of the nucleus. Now, it's actually a double membrane, so there's two layers of membrane here. Okay, the nuclear pore, like I say, is a gap in that nuclear envelope, and that allows molecules to enter or leave the nucleus. Now, it's really important that you don't just stop there and say the nuclear pore allows molecules to enter and leave. You always have to give an example And along with the example, you have to state whether if it would be entering or leaving the nucleus. So an example here, which I think you'll all know, is mRNA, messenger RNA. That's actually formed inside of the nucleus and it moves out of the nucleus. So you could give that as an example. The nuclear pore allows mRNA to leave the nucleus. Now, chromatin, like I said before, is actually the DNA inside of the nucleus. Now, you'll learn more about the structure of DNA at A level, and you'll learn that actually DNA is coiled around some proteins, and those proteins are called histone proteins. And what it does is it allows these big, long strands of DNA to coil around these proteins, packaging it up into a more compact package, which you know as a chromosome. Remember, our DNA codes for all of the proteins that a cell requires. So that includes things like enzymes, hormones, transport proteins. These are the really, really important aspects of a cell. And the DNA that codes for those proteins is only found in the nucleus. So we need to package it up really safely to make sure that it doesn't get damaged, it doesn't get mutated, and those proteins can be coded for as and when they're required. Okay, and then we have the dark spot in the center. So this is called the nucleolus. And I'm sure from GCSE, you will remember, as I said earlier, that you sometimes will find ribosomes present in the cytoplasm of a cell. Okay, well, this is where those ribosomes are actually formed. So they're made in the nucleolus. And ribosomes are actually made from two molecules, one called rRNA, ribosomal RNA, and protein. And they're composed of two little subunits. And like I say, they're responsible for actually manufacturing proteins within a cell. So they would be formed here in the nucleolus, and then they would leave the nucleus via the nuclear pores and enter the cytoplasm, where they could start making proteins. So that's another example of the nuclear pore and a molecule that would leave the nucleus through that nuclear pore. And finally, the nucleoplasm that we mentioned earlier is a jelly-like substance, and it's what the DNA, the chromatin, all these chromosomes and the nucleolus are sort of positioned within. It acts as a medium that they can sit in there. Okay? And it is very much like the cytoplasm, but as you'll find in biology, lots of substances have a different name if they're in a different position. So nucleoplasm, in effect, is cytoplasm, but because it's found in the nucleus, we call it nucleoplasm. You know, actually, the nucleus is even more detailed than that. So what you'll find in the cytoplasm are even more organelles. So you already know about mitochondria. You already know about chloroplasts that you would find in plant cells. And like I said earlier, you also know about ribosomes. So can you see here how that nuclear envelope actually isn't just a single double layer surrounding the nucleus? It actually projects out and forms something called endoplasmic reticulum. And can you see that endoplasmic reticulum has ribosomes dotted all around it? So this endoplasmic reticulum is a continuation of that nuclear membrane. So the endoplasmic reticulum is actually made up of the same membrane that the nuclear envelope is. And this is an organelle that you'll look at later in the course. But because it's studded with these ribosomes, well, then you can guess its function, that it's involved in manufacturing those proteins, because that's what the job of the ribosome is. So the endoplasmic reticulum membrane acts as an anchor for all of those ribosomes. So if we look now at the functions, but A-level style, you can see they're a little bit more detailed compared to what you've learned at GCSE, which was just that the nucleus controls the activities of the cell. So here are 3 bullet points, and these would all be given credit in an exam situation if you were asked what was the function of the nucleus. Okay, so yes, you can see there, it acts as the control center of the cell through the production of mRNA and tRNA. Both of those are responsible for protein synthesis. So when we say the nucleus controls the activities of the cell, in actual fact, what we mean is it provides the code to make the proteins, and it's the proteins that then control the chemical reactions within the cell. Okay. So the second bullet there is about retaining the genetic material of the cell. So the actual nuclear membrane acts as a barrier between the cytoplasm and all the reactions that are happening in the cytoplasm and the inside of the nucleus, where all of the DNA is coiled up, like I said, around these histone proteins to form chromosomes. And the nuclear envelope keeps that DNA packaged and retained in that one space so that the DNA doesn't get damaged. And then finally, and this happens in the nucleolus, the manufacture of the rRNA and therefore the ribosomes that are responsible for that protein synthesis. So you can see the step up from GCSE to A-level from what looks like a very simple organelle in the cell, and actually it contains many more parts, each with their own function. Now you need to be able to recognize the nucleus from diagrams like this. So this is a very simple diagram of an animal cell, but an A-level standard. So you can see it doesn't look like this fried egg diagram anymore. Now, I've actually copied and pasted this from an exam question, so just ignore where it says B and C. The exam question was obviously asking about the structure and function of those organelles, which you don't need to worry about right now, but you can see how I've highlighted the nucleus there. So if you were given a diagram of a whole cell like this, you would be expected to be able to recognize the nucleus, but not just the nucleus. You would be expected to be able to label all those different components we've just talked about. So you can see the nuclear pore there. You can see the nucleolus, that darker spot in the center. You can see where the chromatin is, and you can see the nuclear envelope. Now, the chromatin is the darker part within the nucleus, around the nucleolus. Obviously, any bits that would appear white would be the nucleoplasm, but it's hard to distinguish between the two on this diagram. Now, not only do you have to recognize the nucleus from a drawn diagram like this, you also have to recognize the nucleus from a photograph. So this is known as an electron micrograph. And these are photographs that can be taken using an electron microscope. And it reveals all of the detail there of this real life nucleus. So again, you can see the various components. You can see nuclear pores. You can see the darker nucleolus in the center. You can see the nuclear envelope and the chromatin inside. And like I say, where the chromatin is surrounded by these white patches, those white patches would be the nucleoplasm. Now, sometimes when students are provided with a photograph like this, they make the mistake of thinking that they're looking at an entire cell. And they think that the nuclear envelope is actually the membrane surrounding the outside of the whole cell, and the nucleolus is actually the nucleus. So it takes a while to get used to looking at photographs like this, but this is actually a whole nucleus rather than an entire cell. So if you were looking at an entire cell using the electron microscope, well, then this is what they would look like. So you can see an animal cell on the left and a plant cell on the right. And again, I've highlighted the particular aspects you can see of the nucleus. Now, you can see the nuclear envelope because you can see that boundary between the outside of the nucleus and then where the cytoplasm of the cell would begin. But on these diagrams, it's incredibly difficult to be able to see the nuclear pores. So I haven't labeled a nuclear pore. You can see the nucleolus, the darker spot in the center, and then the chromatin within the nucleus there. And like I say, with biological diagrams, this is often something that you have to either prepare for yourself as part of your practical endorsement, or within an exam situation, you may be asked to label a biological drawing or diagram or photograph like this. And it's really important that you learn how to do that correctly. So can you see throughout this whole presentation, all of the labels that I've used have label lines that touch the feature that they're labeling. And that's really important that you use lines and not arrows in biology, because an arrow represents direction. And we're not showing the direction of something moving here, we're just labeling a feature. So it's really important that you use a ruler, that you use straight lines, and that you use lines and not arrows.